my work and the collaborative effort that we try to put together to address this kind of multi-scale challenging in the field of organoid. I have to say also that it's a bit emotional for me because it has been a long time I attend MIFOBIO and I have always dreaming more or less actually to present something here. So yeah, I'm a little bit emotional here. So sorry for that, but let's move on. So yes, I will discuss today about this huge world actually that started a long time ago. First, as it is education workshop, education meeting here, just a little bit of vocabulary to make sure that we are all at the same page here. So I'm going to discuss about organoids, spheroids, organochip, whatever oid, let's say. So clearly, I encourage you actually to check what has been done by the other GDR on the organoid and this nice representation of the complexification of the organoid here made by Laurine Capdeville, actually. So here we are talking about something that it's more or less in 3D and an object, a multicellular object that actually we want it to recapitulate a certain function, of the original tissue. And we are going to talk about multiscale. So here we are talking about cellular scale, which is between 10 and 30 micrometer, but we are also going to discuss about something that it's much more complex to analyze, like the secretome, like the extracellular vesicle. Here we are in the nanometric scale, but also as it is a very complex system that we call also microphysiological system, we would like actually to be able to know what's happened at a multicellular level. And you will see that actually this multicellular level to address in the field of organoid is not so simple, also because it's a new technology, new method, and it's not so easy actually to address that, even though actually there is some imaging technique that exists already, especially for actually bridge together some multiscale, like for example, correlative light and electron microscopy, or also spatial transcriptomics. So this is kind of things that anywhere you can find it here, so it exists already. The problem here is that we still need to integrate analysis tools actually to be able to bridge all this information across the scales. And once again, here we want to bridge structure with function and with molecular signature. And why is it different or new as compared, or more complicated as compared to other models? Actually, I made a comparison with my colleague, Merlin, that will present something on the zebrafish. So let's call that the historical 3D physiological models used in microscopy. So they are multiscale already, but they are stereotypical, meaning that at the multicellular level, actually, it's more easy, it's very easy, Merlin, to segment, and it's more or less always the same structure that you are going to produce. And why it is such easy, sorry for that once again, but it's because actually the differentiation protocols or pattern is fixed by the species by itself. More or less you put the fish and the fish actually will deal with the differentiation process, and with these species, he will take care of all the protocol of differentiation and growth factor. So everything will look like almost the same between the mother, the sister, the daughter, the cousin, etc. So it's really something that it's predetermined by the species. And also we have a high level of knowledge of that, because actually we have extensive baseline information in this, which clearly is not the case of this new object of interest, which actually it's a mess. It's really complicated, and every single lab has its own protocol and its own interest. So what type of challenge, multi-scale challenges we have here, and what type of question actually we can have using this organoid? So it could start with a very simple question. how many cells per organoid, spheroid, whatever oid, which is not so easy to answer actually. But we can go a little bit deeper in terms of mechanobiology question, like is there any force or mechanical stress within the core of the organoid as compared to the border? And also, for example, the size of the morphology of the cells at the core of the organoid, are they different as compared to the one at the periphery? We have also some questions related to the differentiation process or to the proliferation process, and also to be able actually, and I will discuss that, about that just after that, about the alignment or pattern or ultrastructure that could be formed by this multicellular structure, because we want actually to have a tissue-like structure. And also in terms of mitosis, proliferation, or apoptosis. Altogether, actually, this is really the domain of the high content analysis or screening. And once again, a little bit of vocabulary on that. So the high content analysis is not new. It's something that has, have been described already and defined already. So this is here, I'm going to talk about pipelines to extract high volume of data from images. So you start with images, of course. We are here at MeFoBio. You are going to process your images and try to have beautiful images also, and you are going to extract your results, make some classification and correlation. This is clearly different about the high throughput screening. High throughput screening means actually you have tens of thousands of compounds, of conditions that you want to test, and you want to be able to see which one is the best as compared to the other. So it's completely a different domain and a different vocabulary here. So this high content analysis, probably you know the work of Anne Carpenter lab. It has been done with a very beautiful paper and very beautiful software also that exists, like a cell profiler analyst. So here, I encourage you actually to check about this, all this paper, even if they are coming a little bit old now, but there is a lot of information and actually how to organize your data and how to be able to do some profiling. So yes, we are talking about cell profiler, but also all this cell painting assay, and now with Best Simini lab also at the Broad Institute. So there is several software that allow you actually to perform your extraction of data. So we start with actually classic, let's say classic one, to be able to do some traditional automatized image analysis based on morphology, mathematics and programming. But of course, recently now we have the artificial intelligence, machine learning or deep learning that allow you to go a little bit further, but not necessary. And so there is several tools and software here coming from OME, Napari, Fiji, Pixemina and Cell Profiler, and also I will insist a little bit on that, which will be actually the data mining tool, like the Cell Profiler analysis that has been published by Anne Carpenter. And this will allow you actually to win a Nobel Prize and to get a nice publication, hopefully. So now we are talking about spheroid and organoid, meaning that we are going to talk about 3D a lot and how to high content analyze in 3D. Of course, now based on optical microscopy. First, what we have tried actually and we have developed in our lab is to be able to have something that is compatible with a viable 3D imaging, meaning, a true lab life compatible. For example, you don't want to spend more than 1 hour, more than 1 minute actually to acquire, is it working? It will work. To acquire 3D imaging in multicolor of your spheroid of interest. Then after that, you want to link that to image analysis process, AI-based or not, to be able to to generate your data and to have your segmentation. Once again, you don't want that your algorithm will spend more than a couple of minutes per organoid to be able to segment all your objects of interest. And then you want to be able actually to bring your biological question in the loop, because for now we are talking about microscopy, we're talking about data, but what is a biological question? So here we are going to put back the biologist in the loop to be able to interpret the data that you are going to generate and to have this kind of data mining process that allow you actually to play easily with your data based on a cytometry-like software. And once again, to be able to publish and to have a high impact on the community. What we faced also is that mainly 80%, 90% of the time, all the image analysis needs are based on nuclei segmentation, cell segmentation, or both. Of course, we can have some discussion about that, about the super resolution, single molecule technique, etc. I'm coming from this field, so I'm not going to kill myself here. But clearly, based on what we can see and the volume of demand that we have in the core facility, for example, if you address this two-level, two-scale of segmentation, you already have a lot of work that has been done. So when we are talking about 3D, I guess you know this problem in terms of anisotropy in Z, when we are dealing with optical imaging. So clearly, in terms of Z resolution as compared to the XY, we are not very good. And we don't want to spend too many time, so we cannot spend, 0.1, 0.1 to acquire our data because it's very huge here. And also we, of course, have to deal with this kind of signal loss that you can observe, especially when you are dealing with this big spheroid organoid. So what we think that, and we have published that actually, is that we need to, based on a very simple staining first, and stain the DAPI, stain the nuclei, stain the contour of the cells using actin or whatever. And for that, normally, we should be able to have a lot of data already generated in 3D, which was not done actually previously. So this is our 3 layers that we obtain. So the layers based on the nuclei, the layers based on the contour of the cells, and also the layers of the spheroid organoid. And this will be very important actually to be able to construct your very complex biological question based on that. So I will not go too much into the detail of that because this is not the topic of this session currently, but here clearly we are put together actually AI-based on StarDist, and also classical morphomathematic filtering to be able to have both the cells contour and the spheroid contour. Yeah, and of course we have tried actually here to democratize what we have done and not to be restricted only to one type of high-level, high-resolution imaging, and to be able to apply our network or workflow on, a very large variety, of cells, of staining, and of resolution. So yes, here it's where I'm playing a bit with we are the best of the world. Let's move a little bit further. I just want to show you this big guy actually, because it's something that is very challenging. So this is a primary pancreatic ductal adenocarcinoma, PDAC, stained with nuc blue, and all the images has been acquired here in France and not in Singapore, not at all, at the Université de Lille with Dr. Alessandro Furlan. And as you can see, it's very huge. It's something that it's 0.5 millimeter, and there is a lot of different structure and a lot of different kind of signal also. And in terms of signal to noise, it was really, a huge variety just with one organoid. And actually, our system worked quite well here. As I said, we are the best of the world, like everyone. And we were able to actually answer the first question, which is how many cells in this very huge guy. And we can say that there is more than 3,000 nuclei in this organoid. We went, of course, a little bit further for the second level of analysis, the second, let's say, scale. It's not really another scale, but it's another layer, let's say. And we compute also the cell contour of this huge baby. once again with a 3D view, because here we are not going to talk about 3D, max projection intensity. Don't talk about that with me, please. Then on top of that, as I said, and this is something that could seem a little bit simple and obvious, but actually it's not so obvious. As you can see with this organoid, there is some vesicle in the middle without any cells, and it's a lot of variation, as I said already, in terms of signal. Whoa, it's terrible. But anyway, thanks to this wonderful lady here, Wei-Ting Hong, who performed basic but very robust, actually, thresholding and morphology mathematics on that, we were able to segment and to have the contour of the organoid. And once again, it has been validated on different type of spheroid and organoid. Altogether, we have put all this layer of segmentation in one software, what we call organosegmenter. I think the word is quite good enough to describe what it's for, but it's not limited to organoid. If you have something else, like zebrafish, Merlin, feel free. You can use it, just that we need a nuclei staining, that's all, and a 3D stack, but it could be also a monolayer of cells. I'm open to other systems. I'm not close to that. So this has been done in partnership actually with a private company called Quantitative. And on top of that, it's great because we have the segmentation and we can generate the data. But now let's go with data mining and not piling. Thank you, Gael, for this comparison. Actually, it's really nice also to not just acquire data like that, but try to organize them and to develop something that allow you actually to do a smart or human data mining on that, and to try once again to reconstruct your biological question. So here we have developed two softwares, the organosegmenter and the organoprofiler, to allow you actually to navigate through your data, but also through your images and through your segmentation. So just very rapidly on that proof of concept experiment, why it is so important actually to have this contour of the organoid. Here, for example, we want to test if under osmotic shock, which is a hypertonic medium, we can see a modification of the cellular roundness in 3D, and if this modification of the nuclear roundness has something to do with the relative position of the cells to the border of the organoid. And we were able actually to actually, and we continue to have some story built on this data set, because we are able to gate our data and graphically actually you put very like a cytometry-like software. So you put a gate, but you can have the feedback on the images per se, and you can see where this kind of population of cells are located in 3D in all your batch of organoid. And we did that also, as I said, for the nuclear position related to the border of the organoid, and we were able actually to decipher different kind of effect close to the border as compared to in the center of the organoid. So as I said, we generate a lot of features. So here you have the piling of the data, so a lot of feature, we don't care. I mean, yes, we care, but here the important message here is that we try to organize, we have organized that in these three layers, which is whole organoid descriptor, single cell descriptor, morphological descriptor, but of course also all this intensity and phenotype descriptor, and also this multicellular pattern descriptor. And I will go a little bit more into the detail of that, because actually, as I said, it's not really trivial, and it could be very complicated in this mess of organoid. So the multicellular pattern, here we have decided actually to try something once again that could be a little bit more universal, let's say, and to be able to answer as much as we can to different type of question in terms of organization of the cells close to each other, and to not have any a priori of the structure that you want to analyze. So for that, it's like you are simplifying the question by actually asking yourself around you, where are your neighbors located around you in the 3D space? So more or less here, we are all at the same level, more or less. And we are all more or less randomly distributed in one plane. And this is more or less like you are asking someone that lives in the central business district of Singapore. It's nice, huh? 3D building, very high, very, very beautiful, very shiny. But if you ask each people who are living here, where are your neighbors, they can more or less draw an ellipse, a sphere, around them, because actually they are in all the direction on 3D and around them. Whereas if you are living in Chinatown of Singapore, which is here and not the central business district, which is here, you are more or less in the same configuration as today. You are in a flat distribution, randomly distributed in a flat distribution. And why I'm doing this comparison is that because we are applying this kind of spirit in our nuclei. Now we have the localization in 3D and the central position in 3D of each nuclei of our object of interest. And we can compute actually an ellipsoid fitting around this nuclei, but taking into account the distribution of all the neighbors around each nuclei. And we can analyze the shape of this ellipsoid fitting. If the shape is more or less, as I said, a sphere, meaning that, well, you're living in the CBT. Yeah, good luck. If you are not as crazy as that, actually, you can also have this kind of configuration when actually you have a disk or a frisbee shape, actually, where everything is more or less around you, but on on two dimension, not on the third dimension. Or you can have something that look like a road where you have a train of cells that are close to each other, but aligned in one direction. And we can analyze that and plot actually this result using this scatter plot of prolate-oblate ratio. And so each dot here, it's really one nuclei. So you compute that for your 3,000 nuclei, and you can see the distribution actually of the neighborhood of each of the habitants of your organoid. And for example, in this proof of concept experiment where we are force the cells to have a shape and to have a layer, here it's a cup shape of ECM, so we have this kind of wine, glass of wine shape of this spheroid. So it's not a spheroid anymore, actually. And what we can see is that the spherical distribution of the neighborhood, so meaning this kind of portion of the scatter plot, was only found in the bottom of this glass. Because actually it's normal. I mean, you will say, yeah, you can describe what you see. Yes, I can describe, but I can measure it also. I can quantify it. It's not just describing the things. Here it's really to quantify the things. And for example, if you compare with a tumoroid distribution, spheroid, there is no Q, there is no ECM, there is nothing. We can see that more than 60% of the cells are randomly distributed. There is no organization at all. Great. We have a proof of concept that we can measure it, and even we can classify the different population here. Then you can bring that on our big challenger here, which is our PDAC organoid. And you can also bring back the biologist in the loop. And first, as we have all this kind of interface that allow us actually to also draw on the images itself, we can also draw some gates in the segmentation layer itself, and saying that, okay, I want to know what's happened in terms of organization in this border as compared to the core of the organoid as compared to the bud here. And this has been possible. As I said, in 3D, we can easily draw some regions of interest in 3D. And we can compute directly actually this graph of prolate-oblate ratio. And what we can observe is that in this region of interest, the center part of the organoid, actually, we clearly end with this kind of randomly distributed signature with more than 30% of the cells that are completely, wherever they want to be, they are. So this is clearly something that is very interesting, and we compare that with the border organization, and here we found that actually clearly they are clearly more aligned, and there is this kind of disk type alignment of your cells. As compared to the bud here, so the yellow region here, cyan region. Yeah, it's me for bio. Where actually we can see that it's more the rod type that is present in this region. So now we can clearly quantify what we think it is, and it's not only a qualification of the structure, it's really a quantification of the structure and the organization at a multicellular level. On top of that, we had some other descriptor, like, so here I just finished with a cell-to-neighborhood descriptor, but of course we have compiled the classical nuclei density descriptor, let's say, so the cell-to-cell distance or nuclei-nuclei distance and this kind of crystal distance. So the classical density, once again, but in 3D. On top of that, also, thanks to this fantastic guy, Titouan Poquillon, we have also developed some object of interest analysis, and this is a mitosis challenge that actually we have conducted internally, and also with some group coming from Johns Hopkins to be able actually to detect automatically the mitosis in 3D. And we know that actually the mitosis and the metaphase plate, it's very complicated. So I will not go too much into the detail, but to be able to analyze it, it's really complicated. And of course, I'm doing a little bit of advertisement. So please feel free to come to our workshop tonight at D'Artagnan, where we are going to try to have a description of what we have developed actually with the organosegmenter and organoprofiler software. Okay, so for the second part of my talk, I will talk a little bit as it is a multi-scale approach here. I will talk a little bit about other things and not only optical imaging. I'm sorry for that. So as we said, there is a high variability, and non-homogeneous protocol, and there is multiple cell type, etc. Meaning that why you are using only imaging, optical imaging to analyze your object of interest. There is no a priori scale of analysis here. Everything is interesting. So that's clearly the multi-technique and the high content analysis. Let's go further. I'm not scared about anything. I can add layers and layers, no problem. But here I'm going to use actually an extreme condition as introduced here to actually reveal what is a discovery experiment. A discovery experiment is really something that, okay, let's go for that. Actually, this is a myphobio spirit. It's really the myphobio spirit. You have something in the cell culture room, you have a microscope here, you have another technique here. Yeah, let's see what's happened. This is exactly the myphobio spirit. Oh, I'm happy. Great. So here I'm going to show you how we have extract actually the signature based on phenotypic and cellular morphology. So this is based on optical imaging, but also something that we have done in using volume electron microscopy, so the ultrastructure analysis, and also the secretome analysis, but very, very rapidly on that. And finally, also the RNA-seq and molecular level. And everything is interesting. I mean, from my point of view. Of course, I'm not alone to do that. And once again, just to try to have a proof of concept experiment here and a proof of concept consortium here, and also to try to push the young people and the young team leader to actually collaborate as much as you are, and the youngest here and the young researcher. Try to find someone actually, and it's very easy, who has some common interest, who have a very critical, technical, and very advanced knowledge of one technique, and you can bring everything together. And this is really, really amazing, actually, to work in this field. So this is few of the names actually who have contribute to this aspect. So discovery experiment. So as I said, now we are able to digitalize our spheroid and organoids. So let's say we have a screen, a small one, of 120 organoids here. We were able to compute all the data, all the segmentation, and meaning that we have all these cells. This is a anonym. Here we put all the cells all together. So it's an anonymous pool, cannot say that, of cells here. So meaning that I have completely removed the condition tag on each cell. I don't know where they come from. And I did a classical sense, but that's something that I don't like at all. It's a PCA. PCA, for principal component analysis, is to say, okay, I have all this morphological descriptor. I don't know exactly what I will be able to look at, but I want to see if there is a difference between my condition, if there is a signature. And actually, it was the case. We can see that these F conditions are very high, meaning that there is a lot of cells that seems to have this PC1. But what is PC1? We don't know. There is a lot of things around PC1. But using, once again, the feedback on the images, we were able actually to put a gate here and to inspect back on the images, on the segmentation itself, where are the cells? And what we found is that mainly 99% of these cells are located in the border of the spheroid. And we were able to actually use that as a guidance for supervised filtering. Let's say that I can do this kind of filtering, but just by putting a gate here and separate these two populations. And doing that, actually, I was able to generate exactly the same results, biological answer, using unsupervised clustering, which was a PCA, using the supervised clustering, which was actually the gating that has been applied here. So it was great, actually. For one day, I can understand what is behind this PCA1 or PCA2. So yes, and here it was to say that actually the most affected cells here were located at the border or the external part of the spheroid. And now you are going to ask, but what was the condition actually? And the condition was a little bit extreme, I have to say. So this spheroid has been placed on board of a parabolic flight campaign 2 years ago. They are just in front of me here in the sorry, I miss it, portable incubator just in front of me. And we were very happy because actually we engaged with 100 and 1,000 organoids, and they are all stay in place, and the phone of the friend also. So it was very huge, actually very huge experiment and preparation to be able to bring everything on board. And of course, I want to extract as much as I can of results coming from that. So let's use this example actually of discovery experiment. So clearly we didn't know what to expect about this experiment, but that doesn't mean that we do not have any justification to do microgravity experiment with our organoid. So just few words about that. It has been proved actually that astronauts, and here you have the twin study, are actually in an extreme condition where they are traveling to space. And all these extreme conditions are related to the physiological aging of the astronaut. And so that you have a ton of data about that, actually, that has been published by NASA. Of course, you do not have so much individual, let's say, and subject, because you cannot send thousands of astronauts to the space and to understand what's going on. This is not possible, of course. So what they did now, and what we are doing actually, is to send iPSC cells, stem cells, organoids, to be able to see what's happened in absence of microgravity. I'm talking here about mechanobiology also, not about radiation biology. This is another topic, and we can spend hours and hours also on the radiation part. But here only on the microgravity, it's sufficient. So here, clearly, what has been proven, this is just one example of a paper, that microgravity actually can improve proliferation of stem cells and also the differentiation in cardiomyocyte. So there is still like a lot of things to decipher and to try to analyze, because it's more or less controversial between what we can see at the cellular level as compared to the health of the astronaut. Once again, this is not the topic of today, so I will move on. Just feel free to discuss about that later on with me. So here we are very interesting about that, because we are the MechanoBiology Institute. So we try actually to understand what are the effects of the physical force, on this physiology and disease. And this is, as I said, something that clearly aligns with this aging and disease, because in a sense, when you are not in a good health and when you start to age, like myself, actually, it's more or less an extreme condition, and it's a pathological condition. Okay, so let's move on with okay. Woo! That's really fast. So now we have this description using the optical imaging, and what we want to do and what we have done actually is to be able to see at a higher resolution what is a nanometric ultrastructure of our organoid, and for that we have used SBF scanning electron microscopy. Just very briefly, what is SBF? So this is how we are able actually to generate the data, is that we are using a katana system, so it's a knife that we cut, we image, at the super resolution level, because it's placed inside the EM unit. So it's very powerful. For me, it was like, wow, can do that. And I'm coming from Jean-Baptiste Siberita lab. So super resolution, I know what is it, but here it's really something completely different and completely compatible also. But the problem is destructive. You cut so that after that you do not have anything to image. So that tricks some high limitation also. And you can generate this kind of nice 3D stack that has been acquired. Okay, that's not compatible. That's okay. That has been acquired actually in collaboration with EMU based at our university in Singapore. And we were able actually to have, hopefully it's going to work here. No, of course, that's not a big deal. We are going to generate this kind of images of interest. So you have to imagine that this is a 3D stack. Sorry for the codec problem here. But we are able also to see some very interesting zone of interest here, like this kind of vacuole that appear, but only on the layer, external layer of the cells, and also all the little. Let's try again, but I'm not okay. That's not okay. Let's don't try again. And here, this is some snapshot, and actually there is a lot of events of interest, but clearly we don't know what is it. So many students and postdocs need it here, please. So here we have our volume. Yes, this one is working. Our volume EM acquisition stack, and of course we want to actually be able to analyze it and to extract some data. Same story as previously, AI for segmentation, and here it's an example to be able to have all the nuclei in 3D at this resolution. And we were able to, once again, to import this data in our data mining software to allow the human in the loop process, as I described already. I will not talk too much about that for the secretum, just to say that here it's also something that we are doing also at the secretum level. But to be able actually to navigate through all this level of resolution, actually you need to think upfront a little bit and to have a smart micro devices here that allow you actually to do all these things. And once again here, thanks to a collaboration with my colleague Gianluca Granchi at the Microfabrication Core facility, we were able to design this Quechua tent, let's say, with a top aperture on the top, with a chimney on the top, and you let the cells enter the Quechua tent, and they will agglomerate all to each other. and they are not going to adhere to the surface, and then you can generate an array of thousands of spheroids here, and it's compatible with multi-well plate. Also, as I said, it's completely compatible with a resin embedding that allows us actually to do the EM and also for the secretomomics. So this is something that's really you need to say, okay, I want to do as much as I can of technique, but maybe it's better to think about what you are going to bring in each technique. And this is really important to have, these kind of things. And on the top of that, actually, this kind of microdome are completely compatible with the microgravity effect and hypergravity effect also. And here, just a very huge thanks to Liu Jun, who were able to bring the biostation on board of the plane and to be able actually to follow what's happened with a GoPro on her. It's really multi-scale here, GoPro, joystick and everything, and to be able to look at what's happened inside the microdome. We can see that there is some beads and the spheroid is still in place. And then 2G. And really the 2G is not funny at all. The 0G is funny, the 2G is not funny at all. But we can talk about that later on. So take-home message, and I think I'm good in terms of time. Yeah. So take-home message for this session of multiscale. So multiscale high content analysis recipe. I'm trying to put something here. Analyze broadly, but collaborate widely, clearly. Do not reinvent everything. Try to have something that is smartly connected in terms of data. Once again, not piling, but mining, and to allow the biologists back in the loop. It's very important. Yes, so to keep human in the loop and to help for the visualization, sharing, and that will allow actually the deep biological insight here. And huge thanks actually to all of the team who were able actually to make that happen. So a lot of things has been done. It's not very easy to go for microgravity experiment, as you can imagine. But yeah, sometimes we can have a little bit of fun. But yeah, it was huge. So, and thank you all for your attention on that.
Okay, so then I can start. I have a question about the organoids, and I don't know if some of them can produce a matrix, and if this eventual matrix can pose troubles when trying to analyze with these methods.
So yeah, we are using some extracellular matrix to be able to have a protocol of differentiation of our spheroid organoids. So we bring some collagen, etc. And so, of course, as soon as you bring some material around your cells in terms of optical imaging, it's always a nightmare. But we kind of deal with that. So there is some technique of clearing the tissue, etc., to make sure that we can access the deep volume of the spheroid.
There another question?
Thank you very much. I was wondering, you mentioned the importance of having segmentation of the nuclei, the membrane, the different scales of the organoids. What's your view about new technologies that we use more now for organoids for live imaging, such as tomography, so label-free imaging, and this needs of having the different scales in this type of images?
Thank you for this question, actually. This is really something that currently we are working on that, to have a label-free approach on the spheroid and also to have the live imaging system, because here I'm talking about a fixed spheroid and organoid and staining, but not yet with a live imaging system, because that will add another dimension. It's not a scale, but this is another dimension of analysis, and that in terms of data, it's not so easy actually to add another dimension each time on that. And we don't want to lose the 3D neither, so that will be a real 4D, etc. So
thank you.
Thank you.
We still have time for questions, if you have some.
Thank you, Anne, for this nice talk. So I was wondering, I saw that on your EM picture, that you had some little thin extension of the cells outside of, I mean, on the peripheral cells. So I was wondering if you know what this is, because actually we saw that also by imaging spheroids. So
yeah, this is microvilli because this is HCT116 cells,
Okay.
colorectal cancer cells. So they are still expressing actually the microvilli process, but only when they are free, where there is no cell-cell junction.
Okay,
So
okay.
sometimes there is some vacuole inside the spheroid, I mean some free space. We can see that there is this microvilli. And actually we are developing also an algorithm to be able to track this microvilli and to measure it also in 3D.
Okay, thank you.
Okay, well, I have another one. When you do this kind of analysis on huge samples, how much do you think of segmented cell you have to check to be able to check that the model did what you want?
Yeah, that's a huge question. I mean, that's a very critical question, actually, because, well, yeah, you can come to the workshop. This is also something that we try to put up front, is that there is no ground truth in 3D. Forget about the ground truth in 3D, it's not possible. I mean, of course, you can hire a ton of students, and more or less this is what we've done, actually, but with a lot of respect, of course, to force them a bit to draw as precise as possible the spheroid and the nuclei on that. But it's still 2D, because you do not have the Star Wars holographic system who display Dark Vador in the middle, and you can turn around and draw in 3D. This is not possible. You are drawing and you are compiling your ground truth only in 2D. And so that actually could be a question that maybe a community can address, is that how you can compare the power of your network, of your AI network, to, I mean, and also the precision, where actually you do not know exactly what is the ground truth. So that's a huge question. Yeah, thank you.
Okay, I still have one. And what's the maximum time that you can expose your cell to microgravity thanks to the parabolic flight?
Not so long, actually. That's why also I call that parabolic flight and not zero-G flight, because it's not zero-G, not alone. So we start with hypergravity, with using this small aircraft. It's 2G and 2.5G. That makes a huge difference. I can testify on that, and also all my team, actually. And so you have 30 seconds of 2.5G. So you look at the sky because it's really at 60 degrees. So we have to prove that actually the biostation can work at 60 degree inclination. So I have to talk with Nikon. So biostations are very robust. And yes, and then you have 10 seconds of this hypergravity. It's very long, actually. Then you have 20 seconds of zero gravity. Suddenly you are free and you can move in 3D. It's very nice, but it's only 20 seconds. Then you have 10 seconds back to the 2G, which is still again. And you repeat that 30 times. And you pray to stay alive after 30 times. And to be able to fix the cells also and to make any mistake after that. But yes, it's really, really narrow. It's very small. But yeah, I mean, it's this kind of experiment, it's needed to prove that you have something robust and sufficient to move forward to have a longer exposure to microgravity using other solutions. So this is not the end of the story of the microgravity, but just a proof of concept of everything is working well.